Excellent. Good evening, ladies and gentlemen, and welcome to the SuperShot webinar. On behalf of AcCELLerated Biologics, the RMT Institute, and Forever Labs, I thank you for joining us. I'm proud to present the highly qualified panel to speak to you tonight about the SuperShot. And we do know what it's like to take time away from your practice and your family, and I assure you that the time you invest this evening will yield value. We appreciate it very much. Your attendance and participation are testaments to your interest in providing what's best for your patients, as well as keeping you at the leading edge of regenerative medical therapies. We have a large audience tonight, and as such, the lines will be muted, but there will be a question and answer session prior to the close of this webinar. As your questions arise, please be ready to enter them in the question box or email that you've been sent before. You will have access to this recorded webinar and contact information for our specialists in the event that we run out of time to answer all questions during the session. With that said, I'm very excited to introduce to all of you the CEO of Forever Labs, Stephen Klausnitzer. Good evening, sir.
Hello, can you hear me okay? Okay, great. Thanks for having us here. Really brief on my part, I just want to jump into why we chose MSite and why we chose AcCELLerated Biologics. So when we launched commercially SuperShot, we filed the IP several years back, but we recently launched commercially and we had a number of distributorships that were interested in representing the product. What we looked for was one with high integrity people working with them, good bandwidth out in the United States, and then also what was really great about AcCELLerated Biologics was their continuing training of physicians and staff, in particular their RMTI. You know, there aren't a lot of people in the world that have worked as closely with mesenchymal stem cells, extracellular vesicles in the lab as Mark. He's been doing this for over 20 years. So please stick around to the end. You can ask questions of Mark and the rest of the team. Also on the Forever Labs team, we have Ann Hozeska-Solgat. Ann is our COO, and Ann helped develop the SuperShot technology and filed the IP with Mark. We also have our CMO, Dr. Thomas Nabedy. Tom's been working with us since the very beginning, and Dr. Nabedy was the first physician to be using SuperShot in clinic, so you can feel free to ask him questions about what he's seeing out there. So with that, I'm happy to hand it off to my good friend. Thank you everyone for being here. Here's Dr. Mark Kedekowski.
Thanks, Steve. Steve's voice cut out a little bit for me there, so I'm not sure if it was for everybody else, but he cut out for when I think he was introducing myself. So I'll reintroduce myself. My name is Mark Kedekowski. I'm the President and Chief Science Officer of Forever Labs. I've been working with Steve for several years, and I'm going to tell you a little bit about SuperShot, some of the background story of how we came to develop it, some of the science behind it, and why we're excited to provide now the option to be able to concentrate extracellular vesicles in PRP with the SuperShot process. I also want to thank AcCELLerated for making this happen, and for you for spending the time this evening to learn a little more about it. So I'll tell you a little bit about myself. In 2001, I started getting my PhD in medical physics, and I was working at Henry Ford Hospital in Detroit, Michigan, in neurology research, and we were developing bone mesenchymal stem cell therapies for stroke and other neural injuries. And at that time, back in 2001, we were the first to actually do that in animals for stroke. And I eventually became a staff researcher there, and over the course of my career, became very interested in not only the mesenchymal stem cell, but also the extracellular vesicles they produce, because we found that a lot of the therapeutic value conferred by MSCs was conferred by the extracellular vesicles that they produce. We actually measured that the MSC can produce almost 5% of its mass per day in extracellular vesicles. And these are very potent therapeutic molecules. And we actually started to transition from just doing MSC therapies to using exosomes and other extracellular vesicles from the MSC as the therapeutic vehicle. So in the course of my research with MSCs, I became convinced that they were very valuable to our overall health. And actually, and I became interested in the loss of the health of the MSC niche with age, and became concerned about the loss of my own MSC niche with age, because I was turning 40 a few years back when Steve and I founded this company to bank. When we started offering adult stem cell banking bone marrow. And that's how I became familiar with not just neurology, but the field of orthobiologics, and became familiar with some of the systems that concentrate bone marrow and the cells within, and became aware of PRP therapy. And seeing the PRP systems back in several years ago, I was kind of struck by the fact that these systems were concentrating the therapeutic portion that includes the platelets, but they didn't concentrate the extracellular vesicle fraction, this low density lipid fraction. And I saw, this platelet poor plasma sitting there that was discarded. And when we would isolate in the laboratory these extracellular vesicles, or which includes exosomes, those are some of the most abundant ones, we would spin overnight. We would spin a plasma or cellular culture medium at about 100,000 G overnight. And you just can't do that in clinic. So I became, really interested in, well, is there a way that PRP could not just include this platelet-rich buffy coat fraction, but could you also include the extracellular vesicles? And so started to look into ways that you could do that. And as I might have mentioned, my PhD was in medical physics, and we've developed a way. This is not physics that we developed. This is a kind of a known trick of physics. One thing you can do is create a two-phase aqueous separation. And what you do, and what we do with SuperShot, is you add a little bit of polyethylene glycol and a little bit of high-weight dextran to stabilizers and excipients that are often used in biologics. And by doing that, you can spin it, and then you'll get a soluble fraction with the polyethylene glycol, another fraction with the solubilized dextran, and then below that, the low density lipids, they become immiscible and they'll get expelled in that spin. And you can do this at tabletop centrifuge speeds. And it just takes 1 minute. So that's how we came about. And Ann and I worked in the lab and developed the SuperShot. So we can, just with one more spin, you can add the SuperShot, this PEGDEX solution, and you can spin out the extracellular fraction. You get a pellet of extracellular vesicles, and then you can resuspend that in your platelet-rich fraction. And they're already in your PRP. These extracellular vesicles, they're present in the PRP, but they're just not being concentrated because they're just not dense enough. So, and how many are in there? Well, plasma is very rich in extracellular vesicles, including exosomes. Exosomes are maybe the most abundant. And what we've done is measured what we can spin out of SuperShot. We've done it many times over and sent it to an external lab to validate using a nanosite. And we get counts of, in platelet-rich plasma, it varies between patients, but it counts between 100 billion to 250 billion extracellular vesicles per cc. And most of those, the lion's share, are exosomes. Those are extracellular vesicles that range between 30 nanometers and 150 nanometers. And so that's what, and we're really excited because, like I said, when we were developing these mesenchymal stem cell therapies, what we found is that the MSC was conferring a lot of its therapeutic effect through the exosomes. And actually, I believe, and not just me, others believe, that the exosome and these extracellular vesicles are kind of they're greater than the sum of their parts. And what they'll do is they will, instead of just like a protein where they'll be used as a building block, they're actually instructive, and they actually will modulate the gene translation in the cells that accept them. And they're almost like a endogenous messaging system. You can think of like packets in the internet. They have instructions for what the cells, that uptake them to. And so we're really excited to actually offer now. You can not just have PRP with the concentration of the platelets, but you can also concentrate this potent extracellular vesicle fraction. And so after the talk, I'm happy to answer some more questions, dive deeper into the science. But that's the overview of the SuperShot and how we came to develop it. And now Anne, our chief chief operations officer, and our lab director as well, will kind of walk you through the clinical application of SuperShot, what the SOP looks like in the clinic. To you, Anne.
All right, great. Excuse me. So as Mark mentioned, I'm going to be talking about the actual SuperShot product. So we have a small slide deck to present. So if we could get that pulled up, please. Okay, thank you. So if you could go to the next slide, please. Thanks. So first off, what exactly is SuperShot? So you can think of SuperShot as a way to differentiate yourself from every PRP on the market. And it does this by applying size exclusion to your centrifugation process. So as Mark mentioned, it's comprised of just two simple ingredients. That's polyethylene glycol and dextran. And these are just two long chain polysaccharides that can be found in everyday items, such as over-the-counter medications, eye drops, and vaccines, just to name a few. So when SuperShot is mixed with the platelet-poor plasma of your PRP procedure, it allows for the low-density, lipid-rich biomolecules, such as extracellular vesicles, to include exosomes, to be isolated using a two-phase aqueous method. So as all of you know, much of the PPP volume from your PRP procedure is discarded. But with SuperShot, you're able to isolate the autologous extracellular vesicles found there and add them back to your PRP. And since SuperShot is simply a centrifugation process, it has the same regulatory status as PRP because SuperShot PRP is just PRP. And we've actually received an opinion letter from an FDA consultant stating so. So we like to say by adding the extracellular vesicles back, you create a more complete PRP. So if we can go to the next slide. So I happen to be a Michigan State Spartan, so that makes me a big fan of college basketball. So I like to think of SuperShot as a three-point shot. So point one, as we mentioned, is that it's more complete. And why? It's because of all the autologous EVs that are concentrated and added back. So we know this because, as Mark mentioned, we measured using a nanosite machine, and we found upwards of 100 billion EVs could be isolated per cc of PPP using SuperShot technology. And also there's zero degradation. So with a shelf exosome product, they're usually prepared by lyophilization, and there's a freeze thaw step that can be extremely damaging to biological material. But degradation is a non-issue with SuperShot because you're literally using these extracellular vesicles within minutes of concentrating them. So point one is that it's more complete. Point two is that it's easy. It's a hand and glove fit with the MSite system, and it literally is just a 1 minute additional spin. So what used to take overnight in an ultracentrifuge at 100,000 RPMs can now be done in 1 minute at 4,200 RPM using SuperShot technology. And point three is that it's safe, and this is probably the most important point because it's safe because it's autologous. You are the donor. And we, like Mark mentioned, we sent samples out to outside laboratories for testing and found only trace amounts of SuperShot remains in the injectate. So we're talking micrograms per milliliter. So SuperShot, three-point shot, complete, easy, and safe. So if we can go to the next slide. So what you're looking at now is just a graphic protocol of the process, and each SuperShot kit will come with one of these as a reference. However, I think the best way to understand how it works is to see it. So we've created a 3-minute training video that we're going to play for you now. Oh, there's no sound, so I'll walk you through the process. So basically, this is the step that takes place after the first spin using your MSite PRP kit. This is the SuperShot kit that you'll get, and it includes everything that you'll need for the process. And SuperShot is supplied in this vial. We ask you to mix it vigorously before using. The contents should be like milky or white to make sure that you have a very good mixture before you draw. So using the provided needle and syringe, you're just going to draw 0.5 cc's into the syringe. And then you're going to attach that syringe to the cylinder from your PRP kit. And you're going to draw 4.5 cc's of platelet poor plasma into that syringe that contains the 0.5 cc's of SuperShot. Then you're going to take a separate syringe and you're going to draw off the excess PPP that you do not need for the procedure and leave the volume that you want to inject. So if you want to inject 5, you're going to leave 5 cc's in the column. Then you can attach the 12 cc syringe that comes with the PRP kit, and you're going to swirl the cylinder to mix. Then you can draw the contents into the syringe and set it aside. This is your standard PRP. Then you'll take the syringe that you added the PPP plus SuperShot and reattach it to the column and add the contents. You're going to counterbalance, swirl to mix. And then you'll counterbalance and place in the centrifuge to spin for 1 minute at 4,200 RPMs. And after that's complete, you're going to draw off all the supernatant, so all the liquid you can remove, please draw off into the syringe. And that's discarded. And then what remains on the bottom is a cloudy layer, and these are the extracellular vesicles that you concentrate and use using SuperShot. So then you can take your syringe that has your standard PRP and put the contents back into the cylinder that has the EV layer. You swirl the cylinder to mix. And then you draw the contents back into a syringe. And this is your SuperShot PRP. So that concludes my portion of the presentation. I just want to thank everyone for listening. Up next is Dr. Tom Nabedy, who will be talking about early clinical outcomes he's been seeing in his clinic using SuperShot PRP. As mentioned, he's our CMO, and I think he represents a fantastic blend of both physician and scientist, as I'm sure many of you joining us tonight are as well. So without further ado, I'll pass it over to you, Dr. Nabedy.
Thanks, Ann. I appreciate the introduction, and I think I'm much more clinician than scientist, but have really enjoyed working with everyone here from Forever Labs. Very briefly, I'm a physiatrist, physical medicine rehab, interventional pain specialist who, like many of you, have ventured into the world of regenerative medicine, opened up clinics specializing strictly in regenerative medicine in 2017, and have just been working closer and closer with Stephen and Mark and Ann and everyone at Forever Labs over the past 3 years. Being fortunate enough to be in the metropolitan Detroit area with Forever Labs being founded in Ann Arbor was a kind of a serendipitous meeting between myself and this team. And I can't speak highly enough about them. Not just their knowledge base and their scientific backgrounds, but their passion for the regenerative medicine space and for really doing stuff and doing it the right way. Every interaction I've had with them has just been fantastic. So when they asked me if I'd be interested in the CMO position, it was an honor, and I'm happy to be part of this team. So they're doing some great things, specifically regarding our work on together with SuperShot. It was an idea that Mark had pitched in, and we have been discussing for over a couple of years. And it really took us until this year to really bring it to patients. So I started treating my first couple patients and started with friends and family, just as I did when I started doing regenerative medicine as a whole. And I can't speak to it. It really has been a game changer. It's a different product. Patients respond differently. The initial post-procedure pain doesn't last as long. They're asking for me to increase their restrictions, their activities quicker, because they're feeling better faster. It's something that I'm tracking. I'm part of the Data Biologics Network and tracking my outcomes and tracking those patients. Hope to have a large enough N in the next couple of months where we can have some real science behind that statement that I'm just seeing and observing clinically. And also working with these guys and various different universities and working on getting an IRB approval to have a legitimate IRB approved clinical trial to put what we're perceiving and what I'm experiencing with my patients with real numbers and real science behind it. So, you know, those sorts of things are what we're working on. This product is brand new to the market, and I'm happy to be one of the first physicians that's able to have my patients benefit from it. I don't have a whole lot more I want to say. I want to leave a lot of time for questions. I think that's going to be where the value from tonight more than anything is going to be. I think you've heard enough from all of us. So with that, I'll turn it back over to Brett, and if it's time, we'll open it up for questions.
Thank you, Dr. Nabedy, very much. We have gotten some email questions coming in. I do see some questions coming also. First one, I'll just, what is the regulatory status of SuperShot? To open to the panel.
Yeah,
Mark,
I can take that. Yeah, I just unmuted myself. Sure. It's a really important question. So the regulatory status of SuperShot is that it is a blood product and SuperShot is a process. And so I say that because the FDA outlines that PRP is a blood product and it's exempt from this 361 or 351 kind of, delineation of human tissue products in that it's a blood product. And there's the FDA, states clearly that a PRP is a blood product, and there are some requirements to be a blood product. It has to be obviously autologous, minimally manipulated, has to be used at point of care, used homologously, and the goal was actually to meet those requirements when we were trying to develop a system that could concentrate the EVs. Like I said, the EVs are already in the platelet-rich plasma, but the point of platelet-rich plasma is to concentrate the most therapeutic fraction. And I know you as physicians know that there's different recipes for how you concentrate and for different applications, and different amounts of volume and different amounts of leukocytes. And so what SuperShot really does is says, what we do is you say, okay, let's not just concentrate the cellular fraction or the platelet fraction, but let's concentrate this low density lipid fraction, including the extracellular vesicles. And we basically looked at it, the FDA's guidelines, we said, hey, and it says, hey, you can do this. And one thing you can do though is you can size, and that's exactly what centrifugation is. You can rinse and you can size. And that's what the SuperShot does. The PEG and the DEX are both non-bioactive. They're both extremely low toxicity. That's why they're used as excipients and adjuvants. And then they are discarded in the process, as Anne showed. They're not part of the injectate. So we wrote a position paper of that, and then we sent that to an outside group called Enzyme Corporation, and they looked over it and they issued a memorandum, which we can share if you haven't gotten already, but basically saying, yeah, they believe this is just another PRP process. When we if and when we take SuperShot and we want to incorporate it into a system, then there'll be it'll part of a device, and we'll go down that path. But at this point in time, it's just a modified centrifugation, and that's what we believe, and that's what Enzyme also believes. And so that's the regulatory status of SuperShot at this time.
Brief addition or additional comment there. Perfect summary, Mark. That was my hesitancy in bringing it to market and starting to use it on live patients as well. And getting that position paper was the point where I was comfortable incorporating it into my process and into the PRP process that we use in our office.
If I could just add to, the exosomes are, and all extracellular vesicles, but exosomes, they're therapeutically potent, and they're, we do believe that autologous is key. It's very germane that you use autologous, extracellular vesicles. They are, from what we learned in research, that they, like I said, they're more than just the sum of their parts. They have instructional, they can carry things that include infectious particles like prions, and those are things that are not screened for in any kind of like QA or QC that you're going to get. So it's very important, that we believe that use an autologous product, and that's why we're kind of excited to offer this option.
Thank you both. Some of these questions may overlap a little bit. The next one I received was, how is SuperShot similar or different to adipose emulsification?
I'll take it, sure. So it's similar in the sense that, you're trying to get a he's getting an enhanced therapeutic effect from a biologic. It's different in the sense that the SVF, the stromal vascular fraction you get from adipose tissue, is the result of enzymatic or mechanical degradation of breaking down of the tissue, and then you get this stromal vascular fraction. And this is just simply a centrifugation, so it's just being, based on the density, you're just precipitating them out. It's not kind of a digestion, and that's why we believe that SuperShot and the enzyme as well believes that it's minimal manipulation as opposed to kind of a breaking down of the structural tissue in the adipose tissue.
Thank you. The next question received, does SuperShot concentrate exosomes only?
No, not only exosomes. Based on the data that we've gotten through the nanosite, the lion's share are exosomes, and we characterize that as being between 30 to 150 nanometers in diameter. But often what you call microvesicles, context dependent. There's other microvesicles that can overlap an exosome in size. And then we also, through our nanosite data, we say that some of these, the diameter of some of these extracellular vesicles go all the way up to about 450 nanometers, which can include things like platelet microvesicles. So it does include many exosomes. And there are many exosomes in the plasma, but the SuperShot also includes other extracellular vesicles, which are also known to be in the plasma.
Okay. Look here, next question. How does a patient's age affect the exosome fraction therapeutic effect?
I could probably take that one. Give you a break, Mark.
Sure, thanks.
So I answer that question similar to how I do for any regenerative medicine procedure, whether we're doing a stem cell or a PRP, with or without SuperShot autologous exosomes. Age is a number. Age is not a biologic definition. We're working on getting a better marker for someone's biological age. In other words, how well is the organism still functioning? So I think all of us have patients that are 55 and extremely healthy. We have 55 diabetic smokers who are extremely sick. So whether someone is of a certain age I think is less relevant to how overall healthy they are. And then the same goes for their exosomes. So exosomes is really you are using the autologous SuperShot exosomal EV product is really just an extension of someone's PRP. If they have a good quality PRP, they'll have even better quality PRP if you use the SuperShot.
Thank you, Dr. Nabedy. Let's see, I just have one. I have another listed. Have you performed bedside characterization of the SuperShot exosomes?
So it is something that we are looking at. So it's one of the big passions in our clinic is to quantify what we're injecting. I think to move this space forward, we do have to be able to put a quantification to that. And we're doing that now on a regular basis. A lot of us in regenerative medicine are doing bedside CBC with diff, basically using some sort of a density gradient. So we've done some initial testing with that, with the nanocyte machine, and correlating density gradients to nanocyte quantification. That is something we plan to have for bedside being able to quantify cells not using a nanocyte, but using a density gradient so that we can quantify those EVs.
You know, I might, I could add something to that. One thing we've done in the lab is taken EVs isolated from SuperShot from plasma and co-cultured it with mesenchymal stem cells in the culture, and we have seen there's a dose response. So the more EVs from SuperShot from plasma that we add via SuperShot to the mesenchymal stem cell culture, the faster we saw proliferation. And we also saw a decrease in senescence, in expansion associated senescence, which is kind of the cells aging as they grow and they expand. But what we saw is that if you add these EVs from SuperShot, the cultured MSCs actually grow more readily. And that parallels what we had seen back in the lab with MSC exosomes. But there is a dose response to that. So the more that we add, the more quickly they grow. We got them to almost more than doubling the rate of proliferation of these MSCs using these EVs from the plasma. But there is a curve, and I've been asked by some physicians, why, if you look at the procedure, what we're suggesting is kind of the SOP is, if you have 6 cc's of platelet-rich plasma, then use 6 cc's of platelet-poor plasma to isolate those EVs, and you end up doubling the concentration of EVs in the platelet-rich plasma. In the long run, I think there's probably a lot of potential there to change those concentrations and see if there's optimizations in those different concentrations. But right now, based on what we know are the best knowledge, doubling we believe is an efficacious point, but where that concentration, just like in platelet, in PRP, standard PRP, there's these different I think preparations can have different outcomes, and that needs to be explored fully.
Thank you very much. Again, ladies and gentlemen, any other questions you have, the question mark in the top right of your screen, if you click that, you can type in any additional questions. I want to give you guys another chance to do that. Okay. Okay, panel. I don't see any additional questions coming in on my side. If your questions were emailed directly in as the initial questions started, we will collect those, and they will have answers tomorrow. This session will also be able to be viewed, a recorded version, if you missed anything. And of course, we will have the contact information, as you see on your screen, for any of our panelists regarding what we've discussed tonight. I'd like to thank you again for your participation and feedback. I'd like to thank our panel from Forever Labs. And most importantly, we'd like to thank you for what you guys do for your patients every day. And we sincerely appreciate your time and your participation. We look forward to talking to you soon. Thank you all.
Thank you.
Hi, everyone.